Hello, my name is Rebecca M. Romero, and in this presentation, I would like to describe our experience using ADMET Predictor and GastroPlus in our Computational Drug Design course here at USC. I would like to start off this presentation by describing some of the computational components that we use in our drug design class. So we use the classical docking to try to understand the interaction of a small molecule with its protein target. And then now we've incorporated understanding or using or integrating biopharmaceutical properties using ADMET Predictor and GastroPlus to try to understand biopharmaceutical properties and how that relates to the oral bioavailability of a drug. Initially, we used software that was freely available to calculate some of these biopharmaceutical properties, but over the years, we started to incorporate GastroPlus and ADMET Predictor into our course more and more, especially with the availability of academic licenses from Simulations Plus. GastroPlus and ADMET Predictor have recently become more accessible to educators, and our experience using the software in an educational setting may be helpful for instructors who wish to develop a similar course. We were initially hesitant to use GastroPlus and ADMET Predictor in our course, so we slowly introduced features more and more as we developed this class. Evolution of the class. So in this table, I'm showing you the number of years that we have taught this class, the type of students. So we teach undergraduate, we've even taught PharmDs. In parentheses are the numbers associated with PhD and master students. So here are the students, and you can see the numbers vary. We've taught it in the summer, in the fall and spring, various times during the semester. And the amount of structure-based drug design that was incorporated into the class is shown in hours. So again, the parentheses indicate PhD and master students. So you can see over the years, we've increased some of the time, and then we've reduced some of it in 2017, 18, and 19. For the undergraduate class, we've added more and more clinical information. For the biopharmaceutical portion of the class, I've also indicated the amount of time. And again, there's less time in the more recent years because we're incorporating other topics. And for the undergraduate summer classes, we've incorporated more and more clinical information into our drug design class. And here, finally, in this portion of the table, I'm showing you the amount of time devoted to ADMET Predictor and GastroPlus. So if we look at the initial year, 2010, we only taught undergraduate students, and about 21% of the time spent talking about biopharmaceutics was devoted to demoing ADMET Predictor and GastroPlus. We did not teach any graduate students or any master students at that time. Then we started to teach some graduate students and undergrads, and we still continued to just demo the programs. Again, we were hesitant to use such sophisticated software in our drug design class, but as we became more and more familiar with the program, and there was the availability of an academic license with Simulations Plus, we were able to incorporate more and more information regarding GastroPlus and ADMET Predictor into our drug design class. So you can see in 2012 we used a lot more ADMET Predictor and GastroPlus with our undergraduate class, and still kept our graduate classes to about 20%. In 2013, we continued adding features. Initially, we were just demoing and looking at biopharmaceutical properties. We started adding metabolism and pharmacokinetics and looking at plasma concentration time curves, fraction absorbed, and other properties in GastroPlus. In 2014, we only taught this class to the graduate students, and pretty much we just continued to incorporate more and more features and allow the students to use the programs themselves. In 2016, we went back to teaching the undergraduates during the summer, and we pretty much incorporate a lot of information with ADMET Predictor and GastroPlus into our class. In 2017, we just taught to undergrads, and it was about 60% use of ADMET Predictor and GastroPlus. In 2018, we stayed about the same, and in 2019, we only taught to undergraduates. So let me just describe our course format, our general course format, which includes various active learning activities, a group project, and supporting lectures needed for the students to be able to understand and complete the project. The undergraduate courses have been during the summer, and they have been about 2 weeks in length, with the class meeting daily during that period. The graduate courses are semester long, with 1 or 2 meetings per week. We teach very similar content between the undergraduate and the graduate courses, and we use ADMET Predictor and GastroPlus with both groups of students. However, with the graduate students, we give them more freedom to explore GastroPlus and other software. So let me just describe our group project, which is really the focus of the class. And the goal of the group project is to use structure-based design and biopharmaceutic predictions to develop or improve a drug. So we usually start off with some kind of protein target. Usually it's a protein class. So for example, GPCR or tyrosine kinase or a particular receptor. And we ask the students to look up structural information on the protein and the drug or inhibitor that binds to that particular target protein. And we also ask them to look up various biopharmaceutical data on the small molecule that binds to the protein target. And this is important because when we start to do some of the biopharmaceutical calculations with ADMET Predictor and GastroPlus, we want to be able to compare the experimental data to the predictions. Next, we have them do some biopharmaceutical calculations using ADMET Predictor, simple Lipinski properties such as pK and log P, log D, solubility, and discuss how they might affect dissolution and absorption in the GI tract. So that was pKa, log P and log D, solubility. Hydrogen bond donors and acceptors, rotatable bonds, surface area, all basic parameters that are used in drug discovery to predict oral bioavailability. We also asked them to make an image of the drug protein complex by searching in the protein data bank to find a PDB structure and to analyze the interactions of their protein with the target drug. And then finally, we usually have them do some kind of oral presentation so that we can see how they've managed to gather all this information, and we want to make sure that the students are on track and they understand what they need to do for the project. In the project, we also ask the students to do some docking and to analyze the interactions the small molecule or the drug makes with the protein target, and we ask them to make modifications to the drug to improve affinity for its protein target. So that might include making more hydrophobic interactions, and then we ask them to recalculate some of these ADMET properties when some of these changes are made. And we ask them to think about the consequences of some of these changes. If I make the drug more hydrophobic, which improves some of the interactions with the protein target, how is that going to affect the ability of the drug to be absorbed orally. Will the change make the drug more soluble or less soluble? And is that a problem? We also do some predictions of metabolites. So we try to look at the metabolism of the drug and some of the analogs and ask questions of how that might also affect bioavailability. And then finally, we do some GastroPlus calculations, and we try to look at pH solubility curves, again, log P, log B, D, how that might affect the absorption of the drug in the GI tract, the dose, absorption and dissolution numbers, and relate that to the biopharmaceutical class, and discuss the pH changes in the GI tract and how these properties might affect absorption in the GI tract. We look at fraction absorbed, the amount of drug in the portal vein, the bioavailability, and we try to see if we can relate the properties and the structure of the drug to how it's being absorbed systemically. Is there some kind of connection? And then finally, we have the students do an oral presentation of all their work. So one of the learning activities involves the students using ADMET Predictor. So some of the objectives for this learning activity is having them examine an Excel sheet in which it's exported from ADMET Predictor, and I'd like them to look at the various properties present on that sheet. Also, I'd like them to be able to do a calculation on their own and then export an Excel file themselves. And I want them to be able to understand or explain how a modification to a structure might alter the ADMET properties and the bioavailability of the drug. And some of the supporting lectures involve the types of formats or input files that are used, structure files that you can use in an import to ADMET Predictor. So we like to use smile strings because they're very versatile and very easy to use, and it's very easy to make modifications. So we devote a whole lecture on how to use, or active learning activity on how to use smile strings and how to edit them. But you're welcome to use other chemical formats such as SDF or MOL files. You could also use MedChem Designer, which is available from Simulations Plus to design some of the drug molecules. So basic lectures on pKa, solubility, pH, ADMET properties such as partition coefficients, log D, and a basic description of metabolism and transporters is very helpful. I also have produced a manual that you are welcome to download from this website to help you with some of these learning activities. Here's an example of an Excel file that can be exported from ADMET Predictor. You can also find this file as supplementary data in this paper. I've also added extra information on this Excel file and some descriptions of some of the parameters that you'll find. And if you look at this sheet, you can see there are many different biopharmaceutical properties and various parameters that ADMET Predictor can predict. So we go through that, we look at the structures, and we try to have the students examine this Excel file and try to pick out some of the biopharmaceutical properties that we're interested in. Here is what the sites of metabolism might look like when you do a calculation in ADMET Predictor. The red circles are indicating sites of metabolism or probable sites of metabolism in this example. GastroPlus active learning activities. Some of the objectives include running a simulation in GastroPlus, so the database simulation, and then running a simulation on their drug molecules to calculate some of the pharmacokinetic parameters. The students need to be able to explain all the tabs in GastroPlus and identify them. They should be able to find the fraction absorbed, the amount of drug in the portal vein and the bioavailability. In GastroPlus, they should be able to identify some of the ADMET or biopharmaceutical properties that might affect fraction absorbed or bioavailability. They should be able to explain the absorption, dissolution, and systemic circulation curves. They should be able to identify important parameters in GastroPlus simulation and be able to explain how modification might alter some of the ADMET properties or the bioavailability. The supporting lectures or the background information or preparation for this active learning activity includes, again, SMILES format and basic lectures on pKa, solubility, dissolution, the biopharmaceutical classification system, basic anatomy of the GI tract, and basic pharmacokinetic parameters such as Cmax, Tmax, area under the curve, clearance, and half-life. Again, some of the active learning activities can be found at this website. Here is an example of the GastroPlus demonstration database in which I demo for the students and let them observe how the program works. I explain the various tabs within the program and let them try a simulation. And then we have the students, after trying a simulation in the demonstration database, we look at some of the curves that GastroPlus can produce. So here's the plasma concentration time curve, and we ask the students to try to find a Tmax or Cmax, or where is the area under the curve, or estimate the half-life based on the plasma concentration time curve. We also try to look at dissolution, so the amount of drug dissolved. So this is a dissolution curve. The amount of drug that's absorbed into the enterocytes, the light blue. The dark blue is the amount of drug absorbed into the portal vein. And the green curve is the amount of drug absorbed into the systemic circulation. And we try to look at these curves and try to understand why there are differences between the curves. So for example, the difference between the portal vein curve and the systemic circulation curve is due to metabolism in the liver. We also then go through importing the drug structure. In general, we tend to leave everything as default, and we look at a one-compartment model, and we let GastroPlus calculate the predicted clearance based on any enzymes present. So this is one of the difficulties we have. We sometimes need to adjust this value so that we get a fairly decent curve. So initially, we were concerned about using GastroPlus and ADMET Predictor, especially with our undergrad students. So we wanted to get some feedback. So we created this survey, and here is shown that survey, but it's a combination of 4 surveys. And in the survey, we're showing the students who agreed or strongly agreed. So we asked them questions about did they learn the importance of biopharmaceutics and drug design? Did they learn how important metabolism was in drug design? Did they improve their understanding of ADMET and pharmacokinetics? And you can see in general, they agreed and felt that they were comfortable in these areas. We also asked them, did they feel that GastroPlus and ADMET Predictor was easy to use? So we did not ask that question in 2016 and 2017, but in 2018, we asked them that question. And in general, the students felt that the program was fairly easy to use. And in general, if you use it in default mode, it is fairly easy to use. The content was mostly new to them, so many of them have never had a lot of information on using this type of software, so it's quite new to them. And then we had very good response rates. So that concludes my presentation on our use of GastroPlus and ADMET Predictor in our drug design class. If you have any questions, please send questions to rromero@usc.edu. And if you're interested in looking at academic licenses for a GastroPlus and ADMET Predictor, I have a link here. Thank you.